While Dr. McClung gets set up, I'd like to introduce her. Dr. McClung received her BS and MS degrees from Texas A&M and her PhD from North Dakota State. She joined the USDA Agricultural Research Service in 1991 after 8 years in private industry. Since 2005, she's been the research leader for the Dale Bumpers National Rice Research Center in Stuttgart, Arkansas, where she's developed 16 long-grain rice cultivars and 9 germplasm lines. Dr. McClung has published more than 70 peer-reviewed articles, 4 book chapters, and over 150 proceedings and abstracts for national and international scientific meetings. She is a leader in developing and implementing rice molecular breeding techniques for the U.S. and is recognized nationally and internationally for her research in breeding, genetics, and marker-assisted selection.
Thank you.
Thanks for being with us.
Well, thank you very much. I certainly want to extend my appreciation to the organizers for allowing me to participate in the Baker Symposium. And it's particularly an honor for me because I'm able to share this venue with a former professor, Dr. Fred Miller, who was a professor when I was a student at Texas A&M University. And I still have Dr. Hollauer's book, first edition of Quantitative Genetics, that I spent many evenings with as I was working on my PhD in North Dakota. So it's a pleasure to be here. Dr. Lamke made a comment at the beginning this morning about that funny word germplasm, and I just thought I'd share a story here. This is a picture of the Dell Bumpers National Rice Research Center, and when it was first built 15 years ago, the sign on the front of the building, kind of where you can see it right here, said National Rice Germplasm Research Center. And a couple of years later, when they actually dedicated the building, so it's now the Dell Bumpers National Rice Research Center, the rice community asked to have the word germplasm removed because they weren't sure what it was and they didn't like what it sounded like. So that was the end of germplasm. So what I'd like to do is give you a little bit of background about how rice came into the United States and how that has started with germplasm. And the story goes on that we're continuing to utilize that. So the rice industry actually started here in the 1600s. And this was back in the colonial days. And so rice was first produced in Virginia and was successful and moved down the coast, the Atlantic coast, into the areas of South Carolina and Georgia. And as that production expanded, of course, it was dependent upon having access to labor, so it was very dependent upon the slave trade that was coming out of the Caribbean islands area. We have some anecdotal evidence that in 1685, some rice arrived on a ship from Madagascar in Charleston, South Carolina, and that rice was given to some of the people there, and they started growing it, and eventually it was called Carolina Gold. And that rice became so successful that it is really the basis for the rice industry today. It went into full-scale production by 1700, and in just 20 years, it became the most valuable export crop from South Carolina. And so rice was very important. We usually kind of think of the southeast area that, about the cotton plantations, but really rice was the basis for the economic development of that region back into the 1600s. And so this rice variety, Carolina Gold, was the basis for that. And the reason why it was so important was not only because it was grew well agronomically, but it had excellent grain quality. And that quality was defined as it's a long grain rice variety that when it has a nice appearance to its clean, clear grain, and when it's cooked, it cooks dry and flaky. So very different from like some of the rices that might come out of Japan, which are very sticky rices, or at that time the rices that might have come from China, which were very firm and not as palatable to eat. And so this rice variety was grown from 1600s for almost 200 years. And so I'm thinking that's probably a record someplace, that such an important variety. And in fact, this rice variety is still being grown today, and it's still being grown in South Carolina, but now it's being grown as an heirloom variety for a niche market in terms of they're selling it to the high-end restaurants there that want to promote historically authentic cuisine. So Carolina Gold is certainly has had lasting impact on the industry. So even though rice production started on the southeast coast there, as a result of the Civil War and the loss of the labor pool, a number of hurricanes that came along the coast there, the industry eventually moved over to Louisiana. And so those first growers that were in the Louisiana area were actually importing seed from South Carolina to try to grow in Louisiana. And one of the reasons why rice is grown in these regions is really because a lot of other crops can't. You have high rainfall, very heavy clay soils, poor drainage, and so they're looking for something that can grow in those sort of environments, and rice worked well. But the reason why rice took off was not just because of the rice varieties and that environment, but it was also because of an influx of technology and farmers from the Midwest that came down to Louisiana. And in fact, one of your own former presidents from Iowa College, Seaman Knapp, actually moved from Iowa down to Louisiana. There's a town named after his own hometown in Iowa, which is Vinton, Louisiana. And he was instrumental in terms of attracting farmers and their know-how in terms of how to manage small grain crops, their knowledge about new mechanized ways of planting and harvesting rice. So now that the farmers didn't have access to a slave labor pool, they had to move into a new world of mechanization, and it was because of these Midwestern farmers and that was made possible. So this combination of this highly valued rice variety, Carolina Gold, this very cheap land down on the Gulf Coast because nothing else could really be grown there, and these mechanized, labor-efficient production practices resulted in this new U.S. rice industry being established in the central USA. And by 1900, over 500,000 acres were being grown in this Louisiana, Texas area. And as that production expanded, of course, there was a demand from the farmers of wanting to have access to maybe even better germplasm. And so there was a call to go out and start looking for other possible sources of germplasm that might work in this environment. So as I said, this was the basis for the establishment of the industry, and this is where rice is grown in the United States today. We have right around 3 million acres that are being grown each year. We have this south central region, which is essentially all long grain production, which we call tropical japonica material, or from that gene pool. And then we also have an area in California, which is primarily medium grain rice, which comes more from a temperate japonica pool. And the Del Bumpers National Rice Research Center is located here in Arkansas County of Arkansas. And Arkansas is responsible for producing half of the country's crop of rice. And so you can see that that is the reason why USDA decided to put us there, so we'd be in the center of the business. So as I mentioned, there is an interest in identifying additional germplasm that might work. And so again, Seaman Knapp went out on some exploration trips through Asia, and these were some of the places that were visited by him or by others, and obtaining germplasm and trying to bring that back into the United States to be tested. There was a variety called Honduras that came from Honduras, and it was actually grown in the United States for about 30 years. And so all of these sources of germplasm from around the world were extremely important for even getting the industry established. And so as we look at the germplasm collection at the National Small Grains Germplasm Collection, the NSGC, It's grown to 19,000 accessions over this last century. And you can see during the 70s we had a tremendous increase, and a lot of that was due to a couple of countries kind of opening up their borders to us and allowing us to have some plant exchange. Certainly when the International Rice Research Institute came online in the 70s, they were instrumental in bringing in a lot of, or donating a lot of germplasm to the USDA. So our collection today consists of materials that come from 114 countries, and about a third of the collection, about 3,000 lines of the collection, are coming from the Philippines. Some of that was from the Philippines directly as a source of origin. Others of it would have been breeding materials that came out of the Erie collection. So we look back at when these first introductions were coming into the United States, and this was right around 1900. They were coming from different countries all around the world, and those were the basis for which breeding programs eventually would become established. Bob Dilday produced a work in 1990, and he identified that the southern U.S. rice breeding materials are traced back to just 23 ancestral cultivars, so a very narrow germplasm base. And all of this material is coming from two subpopulations of rice, which is called the temperate japonicas or the tropical japonicas. And so we started off with a very narrow germplasm back in the 1900s. So this is essentially looking at 100 years of our breeding program. At the top here, we're looking at the Carolina Gold and the Honduras and those first introductions that were brought in to the United States. And these were essentially land races, and for a number of years, people were just selecting lines that they had identified amongst those land races, releasing those as new rice varieties, and having those commercially produced. So it wasn't until about the 1920s when we started having official breeding programs established, and they actually learned how to start making crosses. Prior to that, like I say, it was either just selection or actual natural outcrossing that was taking place in the field that they'd identified. But they started making crosses in the 20s, and it wasn't until about 1940 that we started actually getting our first releases as a result of a breeding program. So how many of us would like to have 20 years to get our first variety out, huh? So during this time, what the breeders were doing was, as I said, they're making really crosses amongst those original selections out of those land races. And what they're looking for would be photoperiod insensitive material that was lodging resistant, disease resistant, earliness. Over about a 30-year, 40-year period, they reduced the maturity by about a month in terms of their breeding. They wanted it glabrous, which means smooth leaves, threshability, so there's kind of that fine line between shattering and when it's too hard to be able to pull off the grain off the panicle. So they want it to be threshability and responsive to nitrogen. In 1950 or so, there was a variety that was released called Century Patent 231. It was a very famous variety because the farmers loved it, had great yield potential. But once it got out into the industrial stream, the market rejected it. And that was because it had horrible cooking quality. And so as a result of that disaster, the industry came together and they said, we want to have grain quality evaluation as part of the breeding process. And so that cereal quality laboratory was established in Beaumont, Texas as part of the USDA. And that interaction between cereal quality analysis and breeding started from that point forward. And actually that became a model that has been carried across to other places in the world where no variety is released unless it's gone through a cereal quality evaluation. So during these years, we're essentially just refining this gene pool, and it's getting narrower and narrower. Now, there have been times when the breeders have tried to actually bring in materials from other places and tried crossing with them, and they didn't like what they got out of it. And that was because all those traits that they had been selecting against, it kept popping up again. And so there was just very little interest in trying to bring in materials from other parts of the world. Not only was it challenging in terms of agronomic traits, but they felt like they had essentially got together a very special package in terms of grain quality that was easily disturbed by making wide crosses. So we had about 30 years where there was essentially no introduction of diversity into the breeding programs. However, there's an example that the parboiling industry was taking off. This was kind of as a result of Uncle Ben's producing parboiled rice for the soldiers overseas during the World War II. And they had identified a rice variety that was being grown down in Mexico that they found was producing a very high quality parboiled rice. And so they asked to have that brought into the country. It was brought in 1955. It's a variety called Jojutla. And that variety was then crossed and back-crossed to the variety called Bluebonnet. And that took about another 6 years to get that into a line that they felt that they could breed with. And then that was crossed to another elite breeding line in the Texas breeding program. So in 1980, Nurex was the first rice variety that was released that was actually developed for the parboiling industry. And you can see they went from essentially almost 20 years it took them to develop that rice variety. And the reason for that is again, that these unadapted materials are very difficult to work with in terms of having poor agronomics and other traits that just were not suitable for our industry. But that variety, Nurex, has been the basis for all of the parboiled rice, that is essentially all the parboiled rice that's being grown today. And this rice has a special gene for amylose content, the waxy gene, which gives it a higher level of amylose content than most other long-grain rice varieties. And when this rice actually goes through the parboiling process, it results in a rice that has better grain integrity, less starch solids loss during the parboiling process. So that gene that was brought in from Mexico is the one that you may be eating if you have chicken and rice soup from Campbell's. This has already been mentioned once before, but the Green Revolution came to rice industry in the United States. And so this is another example of bringing in an important source of germplasm. Because of the great success of the variety IR8 that was developed at Erie in the Philippines, that was released in 1966. And Hank Beechel, who was the first breeder with USDA in Texas, he had left Texas, had gone to Erie to work there. He saw what the impact that semi-dwarf gene could have in terms of rice production. And so he took it upon himself to develop, through a series of back crosses, to move that semi-dwarfing gene into a variety that he had helped develop in Texas called Bluebell. And then he sent that back to Texas to Charlie Bollock, who was the breeder there. And he began crossing with that a few years later. And so there was one other semi-dwarf variety released prior to Lamont in Texas, but it wasn't very successful. But then just like 2 years later, the variety Lamont was released, and that transformed the industry. This was our first widely successful semi-dwarf rice variety and allowed yields to really increase because they were able to put on higher levels of nitrogen fertilizer without there being the problem of lodging. This source of semi-dwarfism traced back to Taichung Native 1, which was found in Taiwan. This is a picture of Hank Beechel standing in a conventional height variety in about 1940. And Hank was probably, I guess, a little bit over 6 feet tall. And here's a picture of Charlie Bollock standing in a field of Lamont. And Charlie was probably about 5 foot tall. So you can see the dramatic change in terms of the plant height. So if you want short rice varieties, you need to hire a short breeder, right? suddenly was interested. I was told that when Charlie first came to the Beaumont station, he would go out into the breeding field, that he would go out there and nobody would know where he was because they couldn't see him. So another example of integration of material from diverse germplasm was in terms of dealing with rice blast disease. This is a fungal disease that occurs just about wherever rice is grown in the world, and it can be devastating. There was a source from Vietnam called TTEP, and this was donated to the United States through the ERIE program in 1978. And Dr. Carolyn Moldenhauer and the group at the University of Arkansas began crossing with it. And so in 10 years, she developed the rice variety called Katie. And Katie possesses a gene called the PITA2 gene, which gives resistance to all but one race of blasts that exist in the United States. And that rice that integration has been the source of blast resistance in many lines that have come from it. So I'll also mention that Karen Moldenhauer is a graduate of Iowa State University, and so Iowa continues to give the rice industry a lot of help. So in 1995, ERIE came out with what they call the new plant type concept. And so this picture shows what a conventional variety looked like, kind of very tall and lanky. And then when they introduced the semi-dwarfing gene, obviously reduced it in height by about 2 to 3 feet, and it had much higher yielding potential. But what they decided to do was that they saw that there were some limitations there and some opportunities for improvement. So their idea for a new plant type was to, within this short-statured plant, was to develop rice varieties that had fewer tillers, 3 to 4 tillers, or depending on how you seeded it, get rid of any unproductive tillers. So you can imagine this is, the plant grows and the tillers are developing up to at some of these varieties, they just continue to form. And at some point the plant starts to remobilize the resources to the grain, and some of those tillers are not able to actually set seed. So what they wanted to do was get rid of those unproductive tillers and try to convert that into full grain. So fewer tillers, a lot more grain per panicle. Again, the short plant height, lodging resistance, sturdy stems, erect leaves for good absorption of light, vigorous root system, essentially a short growth duration, less than 130 days, and good disease resistance and acceptable grain quality. Well, this plant type that they were targeting was actually the plant type that has been established in the United States and had been worked on for many years. Our material has been lacking is in terms of this yield potential. We did not have this level of seed production on the panicle. However, we were not going to allow acceptable grain quality because of the disaster we'd learned from in the past, and so that has always been something of high priority for the breeding programs in the United States. So in the 1990s, we started getting access to some of the genetic marker technology. And over the last 10 years, we have worked to develop genetic markers that are linked to some of the major genes that we know are important to the breeders. These have been, again, looking at grain quality, amylose content, which determines whether the grain is going to be soft cooking or firm cooking, gelatinization temperature, aroma. Some of you are probably familiar with basmati or jasmine type of rices. These have a natural aromatic compound, and so that's another trait for some of our specialty markets. We have markers for some of the major blast resistance genes. The PITA gene that came in from TTEP, PIB gene came from another Chinese variety called Teking. PIZ came from another Chinese variety, and several PIK alleles that also came out of China. We also have markers for glabrous, and at this point in rice production, we have probably 70% of our acreage that is being grown to clearfield herbicide-resistant cultivars. And these are a result of mutation breeding, and so we now have markers that we use and provide to the breeders for the presence or absence of clearfield. So even though we had these major genes and markers that worked very well for the breeders, there's still been this ongoing reluctance, and this is being actually provided to the breeders through the USDA kind of at the end of the cycle as part of the Uniform Regional Nursery. There hasn't been much acceptance of using these markers early on in terms of the breeding program. So as we look back at the breeding program, it really wasn't until the 90s, like I was saying, that we had developed some of these markers for these major genes, that marker-assisted selection was actually used to develop a couple of rice varieties, Cadet and Jacinto, using markers for grain quality. And those varieties were developed in 5 years as compared to our traditional method, which was taking either 10 or 20 years to develop. And so that just shows that one can be very effective in one's selection when you have access to that sort of technology. So we've now, with USDA, we are part of the curation in terms of the germplasm collection, 19,000 accessions. We're responsible, even though that material is housed in Aberdeen, Idaho, rice doesn't grow too well up there. And so we grow all of those rice accessions in Stuttgart, Arkansas. And we have, over the last 10 years or so, developed a core collection, which is representative of the whole collection. It's about 10% of the collection. And so that material we have extensively phenotyped and genotyped with, at that point, 71 microsatellite markers. We went from there and developed what we call a mini core, which is just 1% of the collection. And so now we're talking about 200 lines that we're looking at. And that material has been extensively phenotyped and at the time genotyped with about 167 SSR markers. And so we're using that material to then essentially survey this collection in terms of the genetic diversity and how that diversity might be used by breeding programs. So we're still kind of under this mindset that diversity is not that good from our breeding community. And so this is our way of trying to, through association analysis, demonstrate that there is utility in this collection that we've been curating. So we've been effective in identifying markers in germplasm for disease resistance, for nutritional, mineral nutrition. We have a new release that's come out that has weed suppression capability, an allelopathy. And so this has kind of been our effort over the last few years, was really to push the evaluation and the utilization of genetic markers to make this material that's in the collection more useful to the breeders. This is an example of some of the work from Dr. Yulin Jha, who's a molecular plant pathologist at our center. And he is, now that we have these genetic markers for some of these major blast resistance genes, has surveyed the core collection for these markers and has identified a number of them that have multiple resistance genes that can be essentially are stacked and giving one broad spectrum resistance to essentially all the races of blasts that occur in our country. So not only do we have the germplasm, we have the markers that go with the genes. So that was kind of our first step into what we might call diversity panels. And over the last few years, I've had the privilege of working closely with Susan McCouch out of Cornell University and to develop a new rice diversity panel we called RDP1. And this is a collection of some 400 different Oryza cultivars. About a quarter of the material is tropical japonica, which is the kind of material that the U.S. rice breeding program is most interested in. And over the years, the genotyping technology has changed, and so we now have a platform that's gone from 1,500 SNPs that it's been evaluated for to 44,000. And a paper's just been submitted to Nature Communications documenting the use of what they call a Hydra chip, or the high-density rice array, which has some 700,000 SNPs on it. That same Hydra chip has been used to evaluate another diversity panel that's been developed at Erie, and it has an additional 1,300 cultivars. 250 or so of those are from tropical japonicas, and those have also been genotyped with this 700,000 array. And we've just finished bringing in this whole diversity panel into the United States from the Philippines. In rice, we don't have the option of putting seed in your sock and bringing it over the border. We actually have to go through a quarantine process, which takes about 1 year. And so there is quite a limitation in terms of transfer of material and exchange of material. In addition to that, I'm working on another grant with Susan in terms of evaluating essentially a U.S. collection, essentially our entire U.S. pedigree of rice cultivars that have been grown in the United States. And that's also being evaluated with these SNPs. And then working, so I mentioned that the Erie panel was developed by a number of people at Erie, including Ken McNally. And then Brian Scheffler, who's with USDA at Stoneville, he's been instrumental in helping to sequence another set of materials from not only the United States, but also from South America. And then more recently, you may have heard of the 3K. This is another diversity panel of 3,000 genotypes that are being sequenced by BGI in China, and that will have some 2.6 million SNPs on it. So what we've seen is these diversity panels have been developed, they're being phenotyped, and they're being genotyped worldwide. And when we look at the results of this material, what we see is that the Oryza sativa is broken out into 5 subpopulations. We have the japonicas and kind of the indicas, and in the japonicas there's the 2 groups, the temperate japonicas and the tropical japonicas. And then there's a group called the aromatics. A lot of these come out of Iran and the Basmati area of India and Bangladesh. And then we have a group called Aus, which is a particular region in India, and then the indicas varieties. And so the United States material essentially all comes from the tropical japonicas. So this is our little corner of the world. And so when you see these other programs like at Erie or in China, where they're exploring the genome space, they're very interested in the indica material because that's, of course, what is adapted to those regions. So this is kind of, we feel, our area of the world and the area that we're going to be putting most of our emphasis on in terms of exploring. So that was the RDP1 400 germplasm lines in the previous slide, and this is what the new 3,000 accessions that is being genotyped in China looks like. And so essentially what happens is you have the same tree, looks the same, you just get more lines. And it doesn't matter if you're using 30 SSR markers or if you're using 1 million SNPs, it still looks about the same. So these subpopulations are very distinct and are very diverse. So when we look at the number of alleles that are present in terms of the diversity within these different subpopulations, what we find is that, and this is a combination of these 2 panels, the RDP1 and the RDP2, and this is the information that's just being submitted to Nature Communications. What we see is when we look at the number of polymorphic SNPs in these different subpopulations, we find that the indica and the Aus material has more diversity to it. And they also have more of these, what we call minor allele frequencies, that they're unique, both minor allele frequencies and private SNPs, or SNPs that are unique to just those subpopulations. And this, again, is one of the reasons why so many people are interested in that material, is because it is so diverse. However, we have still a lot of diversity over on the japonica side, and certainly plenty of genomic and phenotypic information to utilize as we go forward. So this is an example of a subset, and again, this is some work with Susan McCooch's lab through our NSF project. And this is a subset of tropical japonica materials. They're the ones in blue. And then we're looking at the 12 different chromosomes. And then the red areas are the introgressions that have been brought into tropical japonica as a result of indica material. And we talked before about the importance of the semi-dwarfing gene SD1, and this is where it lands in terms of chromosome 1. And you can see that all of these are some of our more recent rice varieties, are the ones that were the recipients of that SD1. And likewise, the variety I mentioned, T-tip, came from Vietnam and gave us the blast resistance gene PITA. This is located on chromosome 12, and this demonstrates where that introgression is. So those are just some examples of some of the ones that we purposely went out to get the trait and introgressed it into the material. But obviously there's a lot of other introgressions. And what we want to do next, of course, is to understand what the value of these introgressions are, what they're associated with, and how they can be better utilized. So that's just looking within Oryza sativa. We also have some interest in looking at wild species of rice, and there is a great diversity among them also. So this is a publication from Rod Wing's group where he is looking at genotyping, sequencing a number of different species, but the AA genome is the one where Oryza sativa, the cultivated species, comes from. And so these are the other species that are in that same genome and thus are easier to cross with, even though they still get a lot of sterility when you do make crosses with them. So these are the ones that we, as we look to wild species to work with, would be the ones that we would look at first. So looking at our collection that we have with NSGC, we do have some of these species that are present in the collection. And so I've identified here's the ones that have the same AA genome. And we're actually doing a fair amount of work with the rufopogons because those are considered to be the progenitor species for the sativas. And so we're interested in seeing if there have been alleles that have been lost through domestication and that should be brought back into the germplasm for enhancement. Again, this is some more of our collaborative work with Cornell, out of the Cornell laboratory. And what Dr. McCooch has done has looked at not only sativas, but some of these wild AA genomes, and to see what their diversity is. And what we have here are the colored lines are the ones that are actually a sativa varieties, and the black lines are the ones that are the rufopogon or the other species. And so you can see that these wild species overlap some of these same subpopulation structure and indicating which materials would be of interest if you're trying to access them in terms of making crosses. So if we're working within tropical japonicas, what would be the best materials to work with that would be maybe less likely to have a problem with sterility. Susan's group has also done a wider survey of materials from around the world and of rufopogon materials, and has identified that not only do you have materials that are kind of are similar to the 5 subpopulations, there's also tremendous diversity amongst them. And so there's just a huge untapped resource, and this is just in the rufopogons of materials that one could utilize in the future for breeding. So who cares? Why would we want, if we haven't even been interested in working with materials from sativa materials from other countries, why in the world would you want to go to a wild species? And so this is an example of one of the rufopogon donors that has been crossed to a variety that we developed called Jefferson, and Jefferson was being used as the recurrent parent. And essentially all these wild species, only their mother could love them because they don't like to stand up, they shatter their grain, they have small grain, they have poor yield, nothing that you would, as a breeder, would really like. However, through these back-crossing programs, we found, of course, there have been other projects that have demonstrated the great value that these wild species have in terms of disease resistance and insect tolerance and stress tolerance. But in this study that we conducted, again with Susan's group, we identified that they have a yield potential contribution also. And so this is part of the study where we had a series of integration lines. These are back-cross 3 lines. And this is the % difference in yield compared to the recurrent parent, which is the variety Jefferson. And the gray lines are a SIB line that actually lacked the integration we had different yield genes on different chromosomes here. And so we had a comparison of the integration with a SIB line which did not have that integration, and then how that compared to the recurrent parent. And you can see that we're seeing some significant improvement in yield. And these two lines here, 43 and the 219, we're looking at somewhere around 25 to 27% increase in yield over the recurrent parent. And so this was just tremendously exciting that this material that would otherwise, you'd look at it and you'd have no use for it, actually has some interesting gene combinations that can really benefit U.S. rice. So this is looking at a graphical genotype of some of these back-cross progeny, and just as an example for the integration on chromosome 2, here we had the back-cross 2 F2 population, this line number 43, which was identified, and here's the size of the integration. And then some further back-crossing was made and selection with an idea of trying to reduce the size of that integration to try to just get down to the area of the chromosome that would be of most pertinence in terms of yield. And so what we found was kind of interesting. So this is where we're looking at the yield potential here on this axis. And so here's the variety Jefferson, the recurrent parent. And in black are the original integration lines, which were BC3s. And then the gray lines are ones which were an additional back cross, or BC4. And so this is for the yield QTL on number 2, chromosome number 2 here, and on chromosome number 6. And so what we find is that, the idea was that with further back crossing, we would probably increase the yield because we would be getting rid of integration that were probably unadapted and not of benefit. But what we find is that actually that was not the case, and some of these integressions, you might say the other integressions in the background, seem to have a significant positive effect. And so, as I mentioned, here's the recurrent parent Jefferson. Here's the variety Presidio, which is a derivative of Jefferson that was released several years later with its yield potential here. And you can see that these integration lines are approaching the yield potential of the new Jefferson, which was released several years later. These are two other rice varieties, Trinas and Cocodrie, which are also widely grown in the southern U.S. And so again, these integressions are able to essentially take an old variety and move it forward dramatically to a level that's competitive to varieties that were released maybe 10 years later. So I think this demonstrates not only the value of these wild species in terms of disease and insect and stress tolerance, but also in terms of yield potential. So where do we go from here? So first of all, these development of these different diversity panels, I think, is our first big step. They reveal the genetic diversity, because we're looking at this material not only phenotypically, but genotypically, and also just working with these wild species and finding out just how relevant those materials are in terms of varietal improvement. And so as a result of some of these diversity panels, there's increasing interest to expand the collection, fill in gaps in terms of our germplasm, and we're exchanging some of these diversity panels. For example, we just brought in the collection from Erie, the 1,300 lines. Recently, we brought in a core collection from Brazil. Brazil has received our entire collection, all 19,000 lines. And so I think having that sort of exchange is going to be very important as a global community starts to look at this material. And also by having these markers, of course, we can scan the material, look for accessions with novel alleles that we may have lost in the process, and try to bring those back into our program. And having these databases being made publicly available is just critical because, for example, the new 3,000 genome diversity panel, since it takes us at least a year to bring these materials through a greenhouse, and that means that you have a greenhouse that can accommodate 3,000 different lines, we're not really that interested in bringing in these large collections. But by having access to the genotypic information and the phenotypic information, we can utilize that database to identify if there's subsets of it that we do want to bring into this country and start working with. So, as I mentioned, so we're looking at this material and identifying and integrating novel and desired alleles. Also, we want to be able to look at the germplasm that has maybe unique combinations of genes that can be used for breeding stock, kind of like the blast-resistant materials. And I'm very interested in exploring the genetic diversity that is within the tropical japonicas and just to try to enhance our U.S. breeding programs without actually going to the point that we're actually digressing in terms of improvement. But then there's also the interest, of course, in trying to maximize the diversity that is between the different subpopulations. Right now in the United States, we have about 30% of the acreage is being grown to hybrids, rice hybrids. And so knowing and understanding the heterosis, and that's coming from crosses between indica with a japonica is where you're going to get that level of heterosis. And so these hybrids are having about a 15 to 20% yield advantage over conventional cultivars that are being released. And so this will give us another way to explore that. So as we look to the future and what are the challenges for our industry, one, we're losing some of our export market. 50% of our crop is being exported. And over the last few years, we've been losing some of that market, particularly to Central America. And as I mentioned, the United States used to be the leader in the world in terms of the grain quality of the material that we have produced. But we've been either losing on that market or we were backing off on that market, and as well as other countries have dramatically improved the grain quality of their material, and so we're having it more difficult to compete in that export market. One of the biggest reasons why we're losing that market is due to a factor called grain chalk. And this is, rather than having a nice clear grain, you have a chalky, opaque center in the grain. And we know that there's a genetic component, there's certainly an environmental component, but we also realize there's been studies that have been done that have shown that what we're seeing over time is an increase in terms of the nighttime temperatures. And we think that that is stressing the plant, and so that rather than able to actually really fulfill the enlarging grain's need in terms of starch, there's actual air spaces that are being not filled in the grain, and so that's causing these chalky, soft areas in the grain. We're also seeing a dramatic decline in irrigation resources. Texas has gone through several years of drought, and to the point that sometimes they're just told, I'm sorry, we don't have any water for you. Unless you have your own water, you can't grow rice. California is going through the same thing. They're not having enough snowfall in the mountains, and they are under water restriction for rice production there. There's areas within the state of Arkansas that are where you have the highest acreage of rice, and these are called critical groundwater areas because the depth of the aquifer that they're pumping from has gotten so low that they cannot sustain rice production. This is the biggest issue that will determine whether rice is produced in this country in the future or not. We also, of course, see changing diets. There's a lot of diabetes in our country, and the rice industry goes, well, how are we going to get people eating rice if everybody tells them not to eat white stuff? And so we're looking at ways to improve the nutritional quality of rice, whether that's through the brown rice itself, or actually developing rice varieties that have higher resistant starch, as Fred Miller was talking about. And of course, as you increase resistant starch, which means it's not digested in the gut as much, it also changes the sensory quality of the rice, and so the rice becomes firmer. And so what we want to do is have enough resistant starch that you don't have a problem with diabetes, but it has the texture that once it's cooked, you still enjoy eating it. A changing climate, as I mentioned, we're already seeing some effects of this in terms of our nighttime temperatures, and so we need to develop rice varieties that are going to be climate resilient. Global warming, as everybody knows, rice is generally grown in flooded paddies, which results in a lot of methane emissions. And so, Fred, you're looking at places in Germany where they had trying to capture that methane. Just come to our rice fields. We've got plenty of it. So this is going to be of concern to future society in terms of what is the impact of rice production in terms of global warming. We have done some studies recently which have shown that rice varieties differ in terms of the amount of methane emissions, and so we want to be able to understand that better and to utilize it in breeding programs. So this is our staff at the Del Bumper Center. I'm, as they say, just a breeder, and I work with all these wonderful people that have great expertise. Dr. Eisenga, who's working with the wild species crosses. Ming Chen, who's working on nutritional quality in rice. She's looking at antioxidants in brown rice and resistant starch. Dr. Rolf Bryant, who's looking at processing and parboiling quality. Dave Gailey, who's looking at allelopathy, weed competition using rice varieties. Shannon Pinson, who's doing QTL mapping with mineral content. Yulin Jha, who's looking at blast and sheath blight resistance. And our two newest hires, Angela Baldo and Jeremy Edwards, both who have their roots in Cornell, have just joined us and are bringing bioinformatics expertise to our group. And I'll just say we have 3 vacancies, and if we ever get to the point where we can advertise them, I hope some of you folks might think about coming down to Rice Country. So with that, I'll close, and I appreciate, again, being here. As I mentioned, Carolina Gold was the beginning of the story, and it is still being grown today, so that just shows the resilience of some of these rice varieties in terms of their importance to our industry, and we look forward to continuing to utilize and mine that material. So thank you very much.
All right, we do have some time for some questions, so please just use the microphones and go ahead.
It's starting to feel like a tropical field in here, isn't it? Kind of warm. All the after lunch crowd. Good. I left you speechless. Yes, sir. You, Dr. Yu. So let me make sure I understood. When we talk about how to translate essentially SSR markers to how many markers we need from SNPs? Yes, yes. Yes. I didn't get that very last part. Oh, high-density stamps, okay. Okay. So the question was, we had a couple of different maps, one with just 36 SSR markers and one with 700,000, and the map didn't change much, and so what do the people that are doing the SNP analysis say about that? Guess what they said? We need more markers. Which is what they've done. So now we're going through, sequencing of the material. And, for a person that walks in the field, I'm like, okay, when are they going to get over this? And it's not about that. But I've come to understand that there are reasons why we need to have more and more SNP saturation. And particularly when you see those big branches of the Oryza sativa tree, there's a lot of linkage disequilibrium. And so how are we going to break up that linkage disequilibrium that are in these different 5 subpopulations? How can we do that, and how can we do it the most effective manner? And having that marker separation or marker density is going to be more and more important. So, God bless them. Let them continue to do more markers. We'll do the best we can to utilize it. And, that has been one of the things, even in our group, and I want to say this, we're in Stuttgart, Arkansas, in the middle of a rice field. We're not at a wonderful campus like this with lots of intelligent young people just anxious to get out there and do something. And so we don't have the sort of resources that some of these university campuses have to explore some of these questions. And so we're just now getting to the point where we have people with the bioinformatics expertise that can really make this material, this information more meaningful to us. So I'm very excited about it, and I think there's people that their whole life is just markers, and I appreciate that. But we're just going to do the best we can to utilize these materials as they become more available. And I've already forgotten your second question. That's good. Okay. All right. Thank you.
I'd like to ask if are you doing transgenic research in your breeding program?
Okay. So are we doing transgenic research? And the answer is no. The industry has been adamantly clear that they do not want GMOs in the rice, and that's primarily because of the European market, which it's not the biggest market for us, but they're a significant market for our exports, and they are very clear they want GMO nothing. And so a few years ago, there was actually, there are some programs that are working with GMOs, they're private companies, and they got a little bit of it kind of slipped out and got into the rice stream, and it essentially shut down the market. And so the problem was that it was an event that had not been released, although it was no different than really another event, but it just really had a tremendous damaging effect. And so as far as the industry is concerned, they're not interested in us pursuing GMOs because of their fear of what it might do to the export market. However, they are also aware that there's a lot of other countries that are working on transgenics, and so they're supportive of someone doing the research. They just don't want it being done in rice country. And so, that day will come when maybe another country makes a big breakthrough, and they're going to want to open up the door and have us try to utilize transgenics, but we are not doing any of that right now. And my thinking is there is such a world of genetic diversity that we have not explored, and that is really our mission at Dell Bumpers, is to just utilize that natural genetic diversity to the best we can. Okay.
We did have a question from our webinar. So David asks, why is the glabrous trait valuable?
Why is the glabrous trait valuable? So glabrous is a smooth leaf, so the plant can have pubescence essentially all over it. And we do have a few varieties that are pubescent. So what happens is when at harvest time, that pubescent hairs, they actually break off, and they're an irritant for humans. They're also tremendously abrasive in terms of farm equipment. And so people that do harvest pubescent varieties in the United States with mechanical equipment, they find that their machinery breaks down quicker and actually wears out. So even though you find pubescence in most other parts of the world, that is a trait that we try to select against just for that reason. Okay? All right.
All right, well, if there's no more questions, let's thank Dr. McClung one more time.
Thank you.